So I've changed my title a little bit to be a little bit more comprehensive. I want to discuss sort of combating infectious diseases and just one path that I've taken with many collaborators to generating novel resources. Um, let's see here. My path so far is kind of unique and did start off at Yale. Uh, I started off in upstate New York as a biochemist at Russell Sage College. I actually started a PhD program at Yale in the microbiology program there, but unfortunately decided to join a lab that was relocating to England. And so I left with a master's, but restarted my PhD at the University of Cambridge, specifically at the MRC Laboratory of Molecular Biology. And this is with a Yale alum, Professor Yorgo Modis. And then I kind of got hit by a triple whammy, which was formative in my sort of academic experience. I then moved to another group in Cambridge that was also a joint collaboration with the University of Sao Paulo in Brazil and involved quite a lot of trips between England and Ribeirão Preto in Sao Paulo with the research institutes there. And then I was struck by a couple more interesting things, some more COVID, and then another international move. And now I'm working as a scientist one for platform biology at Big Hat Biosciences, which is a machine learning and data-driven antibody drug discovery company. And I work largely in assay development for drug discovery. Obviously, things have been exciting, and so I just don't know what I'll do next. But I'm very committed to a career in combating and developing resources for addressing viral emergence. So my doctoral work was mostly focused around the structure and function of viral glycoproteins. And I spent many years studying the evolution between a 20 million year old human hookworm and a retrovirus within its genome and its relationship to modern tri-segmented phlebiviruses. With collaborators in our group, and this is again with Yorgo Modis. Oh, my emphasis here is that this was 6 years of doctoral work, which took another 18 months to get published. So if the timelines I just look back on now and I find remarkable because it's such a long haul, really. But we used X-ray crystallography and a bit of cryo-electron microscopy to do structural characterization of the Acinetobacter atlas virus. We used protein mutagenesis and some biophysical characterization to study mechanisms of this GC-mediated cellular membrane fusion. So this is a class 2 fusion protein, which is very closely related to dengue, Zika, Rift Valley fever virus, most of all. And we combine this with a bit of phylogenetics on hookworm endogenous retroviruses and their relation to modern class 2 fusion proteins to assess the dynamics of virus evolution. And while I'm not going to talk about a lot of this today, I did have some takeaways, which were that retroviruses can be really remarkable genomic fossils for modern viruses. And there's lots of genomes out there for scavenging and lots of retroviruses that have captured aspects of viral evolution. We found that there was uncanny conservation of this glycoprotein structure and function down to the lipid membrane determinants, the pH sensitivity, its sort of functional intermediates through 20 million years of when the retrovirus was integrated into the Acinetobacter hookworm to the modern phlebiviruses that are circulating, most of all Rift Valley fever virus, which is prevalent in Africa. And that finally, these genes that encode viral glycoproteins are tremendously promiscuous like plasmids. So the sort of envelope gene that you would see in a retrovirus is very similar to the M segment for this family of viruses. And so that these are passed around pretty frivolously in evolution and they're useful. And so they get conserved remarkably despite that sort of transmission. I learned quite a few lessons in my PhD. It was a very long one, and it was to question what you think you know and check for yourself. There was about 2 years here where I worked on a misannotated genome and a protein similar, a homologue of this that was a splicing mishap. And so it didn't express, it never worked well in my hands, and there was a reason for that, but it took a lot of digging to get there. I learned not to let others tell you what you're capable of. I set out to do a really big project in the beginning, and in the end, we did succeed. But I think had I had the support in the beginning, I might have gotten there sooner and been able to celebrate it a little bit earlier. And to share your ignorance and expertise and enthusiasm with others, just because there were countless times in my doctoral studies where a senior virologist or someone would come in and engage, and it really transformed my personal experience. And so um, I think we really need more of that. And speaking with each other and celebrating each other's research is really helpful in um, generating good scientists. So now I'll focus mostly on my postdoctoral work, which is unpublished. It's up on medRxiv, but it's in review also at EMBO Molecular Medicine. So it should be available to all of those of you that are interested. And this was 3 years between the University of Cambridge and the University of Sao Paulo. And then still 18 months more before any sort of written record of this has come out, despite there being an active and current Oropouche virus outbreak. And so we were working before this outbreak to develop low-cost diagnostics, candidate vaccines, antivirals, and lab reagents for this very much neglected tropical disease, which is insect-borne, just like the phleboviruses I studied in my PhD. So with collaborators from Brazil, we used recombinant proteins, viral antigens, GC spike, and nucleoprotein to assess immune responses to virus infection. And this is something that had never been done before because mostly this virus circulates in South America, in Brazil, in the Caribbean, in Peru, and there just hasn't been a long history of research into it. So this is quite novel at the time. With those antigens, we're actually able to discover that the current Oropouche virus outbreak is actually likely 5 times larger than PCR testing indicates. And exactly what's going on here is unclear, but our leading hypothesis is that there's very acute viremia in the bloodstream. So PCR testing is not the ideal diagnostic for capturing the true size of the outbreak and the number of patients that are clearly have antibodies, IgM, against this insect-borne virus. We use those same viral antigens, the GC spike protein and the nucleoprotein, and tested them as novel or candidate vaccines in a mouse immunization trial in a prime boost mechanism followed by a challenge, and found that these antigens elicited a protective immunity, and we really demonstrated the potential of a novel vaccine for this virus. In addition to that, we used these recombinant antigens and immunized llamas to produce single domain antibodies, which are sometimes called VHHs for variable heavy, or trademarked and called nanobodies. So you might have heard of all of them, but essentially they're a very small version of an antibody with just the variable domain, which makes them very useful tools for any pathology, but especially for virology. Using these antibodies, we characterized numerous versions and a couple different formats and developed a sensitive and specific diagnostic ELISA for Oropouche virus detection, because at the moment there is no clinical test for Oropouche virus. It presents very similar to dengue virus and or Zika virus. And so there's a lot of misdiagnosis in South America, and most cases go unmonitored without this sort of low cost and accessible diagnostic. The reality is, in that clinics where Oropouche virus is present in the Amazonas region, there's no capacity and or resources for PCR testing. So that just isn't happening on the regular. Over the next couple of years and with collaborators, honestly, across Brazil, in Sao Paulo, up in Manaus, we were able to validate this diagnostic using clinical samples from both historic and current outbreaks. So on the left here are actually numerous patients, clinical samples that did not have dengue, did not have Zika, but had some febrile, potentially viral illness. And using our diagnostic, we were able to show that a good amount of those were actually Oropouche virus and just were unconfirmed. So there's a huge number of non-diagnosed cases sort of circulating unmonitored. And then recently, in I think 2023 into 2024, we had the most recent outbreak of Oropouche virus, which is spread across South America. And we were able to validate this diagnostic with a series of samples, again, from this outbreak, where the green points are PCR positive Oropouche virus samples, and the others are PCR negative. And so our diagnostic was able to detect that in at least two of the patients. Using these small nanobodies or VHHs, we were able to actually make some very useful laboratory reagents to study Oropouche virus infection because there's very few commercially available antibodies and or serum samples to study viral infection in vitro. And so we discovered using these fluorescently conjugated nanobodies that the current circulating strains of Oropouche virus have very distinct cytopathology from the laboratory strains, which really emphasizes the need to invest in this sort of research because we think we know what we're looking at, and the reality is what's out there is already evolved and quite different. And then finally, we also tested these reformatted VHHs as candidate antivirals in virus neutralization experiments. And so despite the differences in their cytopathology, these nanobodies against the glycoprotein were potently neutralizing for both lab strains and the currently circulating strains of Oropouche virus. So this work was really fulfilling and quite a gargantuan effort between a lot of researchers in different universities with lots of preparing reagents, traveling to South America, doing experiments, bringing colleagues back and sharing our work worldwide. Our scientific successes were mostly producing the first low-cost clinical diagnostic for Oropouche virus. We identified candidate vaccines and antivirals to combat infection, developed some very versatile laboratory reagents, and identified some differences between current and laboratory strains of Oropouche virus. Some of the lessons learned here were that, as you all can appreciate, climate change is really driving broader virus transmission, and this is largely due to expanding geographical ranges of vectors like mosquitoes and midges. And we also learned that biosurveillance is really critical to combating this infectious disease, which was really demonstrated in the recent outbreak. And finally, that rapid resource development, specifically for neglected diseases or neglected tropical diseases, is really possible with commitment, investment, and international collaboration. So there's a question of like, then why would I move from academia to industry and specifically big hat biosciences? And that's because I really believe in machine learning driven platform technologies. This rapid scaled data driven reagent and or antibody development could be really useful if it were applied to areas that need it. And I think that global public health really needs collaboration between academia, industry, and government because we're facing climate change and habitat loss, globalization and forced migration of both people and animals, and a rapid loss of healthcare infrastructure. And so I really think we need infectious disease experts more than ever before and advocates for science and public health with real world solutions in the correct sort of response times, and that this technology could really do that. Not that Big Hat is currently working on any infectious disease targets. We're a target agnostic company and do a lot of research, but I'm hoping in industry I'll be able to convince people to shift their focus to maybe some more altruistic work. And so I've put some contact information here, but I do want to emphasize that all of this work was, my PhD work was led and supported by Professor Jorgo Modis, who relocated from Yale to Cambridge, and that the postdoctoral work was with Professor Stephen Graham, Eurico Arruda, and Luis Liberci da Silva, all of whom I'm very grateful for. And so if you have any questions or if you want to reach out to me afterwards, I'm really looking forward to speaking with all of you. So thanks.